Well, hello everyone. My name is Shalyn McCoy, and on behalf of the Foundation for Conservation of Atlantic Salmon, I just want to say welcome and thanks for joining us for today's webinar. I know some of you have attended our past webinars. I'm glad to be able to finish this series here with Dr. Ian Bradbury. Dr. Ian Bradbury, he's a research scientist with the Department of Fisheries and Oceans, originally from Newfoundland, and he completed his PhD in 2007 at Dalhousie University and started with DFO in 2010. His research uses genomic tools to inform the conservation and management of marine and anadromous species from throughout North America. I just want to say a special welcome to Ian. He's also one of our volunteers on our Scientific Advisory Committee and part of the Foundation. His talk today is going to be on eDNA, which is a tool that can be used to better understand Atlantic salmon and other species, but I am not the expert on that one, so I will let Ian do his presentation here today. Before we start the presentation, we do have French translation available to everyone today. So if you go in your chat, all the instructions on how to log into that, and you'll be able to do that and just choose the language of your choice. So we'll start the presentation. So Ian, it's all yours, and looking forward to hearing it.
Great, thank you. Well, thanks folks for joining. What I'm going to do this afternoon is provide kind of an introduction or a little bit of a primer on eDNA and its use in conservation and management. And then I'll talk a little bit about some of what I think are some of the better examples from the literature, and then some examples from my lab in how we're using environmental DNA or eDNA to inform the management of anadromous and marine species. And I have my email up here, so if anything isn't clear or if you have any questions, please feel free to drop me a line after. I'm happy to chat. Before I start, I always like to acknowledge that a lot, especially with the examples that I'll talk about, it really is a team effort. So that involves a lot of DFO staff from Newfoundland, shown here in the field. It also involves graduate students and postdocs in my lab, as well as we work very closely with the Center for Environmental Genomic Analysis in St. John's, and they do a lot of our library prep and sequencing of our environmental DNA. So special thanks to all these folks. So what I'd like to do in the talk then is kind of broken into these 5 bits. Again, because eDNA is a little bit of a newer technology, and I know oftentimes these webinars have quite a diverse audience, I'll start a little bit with a primer. What is DNA? What is environmental DNA? How do we actually sample it? What types of analysis do we do? That sort of thing, just as a general introduction. Then I'll talk about, as I said, some of the examples from the literature that I think highlight the potential and some of the limitations of the technology. And then lots of examples from work done by grad students and postdocs in my lab. And then I'll summarize quickly. So probably don't need much background in terms of what DNA is. It's a self-replicating material. It's present in almost all organisms, and it forms chromosomes. So it is the carrier of genetic information. Now, what's relevant for us here is that it's found basically in 2 places. It's found in the nucleus of the cell, where we have the chromosomes, but it's also found in mitochondria. So mitochondria, kind of the engine house of cells, they have their own genomes, own DNA. So there are essentially 2 types. There's nuclear DNA and there's mitochondrial DNA that I'll talk about today. And of course, there's a very long history of using DNA differences among individuals, populations, or species to inform conservation measures and conservation action. So what is environmental DNA? Well, as organisms, be it fish, Atlantic salmon, or whatever, move through the environment, cells are excreted or sloughed off in the form of mucus, and this contains DNA. So environmental DNA is essentially the DNA that is lost to the environment as organisms move through. And again, that can be mucus, it could be excreted, it could be all sorts of different types of cell types. And what's happened over the last decade, or probably a decade or 15 years, I guess, is that there's been dramatic improvements in how we read DNA. So changes in technology now allow us to read massive amounts of DNA very cheaply compared to 20 years ago. And then I always show this plot. It's probably getting a little old now, but this plot just shows the cost of sequencing a human genome and how it's really gone from millions of dollars to less than $1,000 in recent years. So that just illustrates how the technology has changed so much. And why this is relevant is this means that we now have the ability to really sift through the haystack of environmental DNA and really read a lot to pick out the bits that are relevant to us. And this sort of new power or new sequencing power really has spurred on a little bit of a revolution, I guess, in biomonitoring. And it started around 2000, 2005, 2008. So here's a figure from a recent review by that came out last year, I believe, and it just highlights just the progression of the field, where we started out with the first sort of detection in aquatic environments, and then we moved to invasives, and then sort of in 2012, we had the first sort of comprehensive eDNA study on marine fish, and then more and more sort of uptake and application. So eDNA is rapidly developing as kind of a preeminent biomonitoring tool with unprecedented time, efficiency, and sensitivity. So as you'll see, and I'll show some comparisons, but essentially what we're doing is, because we're essentially taking a water sample and filtering and focusing on the DNA present, it is very quick, it is very time efficient and very cheap to sample. Compared to traditional methods such as trawling, netting, these other sorts of approaches that require extensive infrastructure and staffing. Of course, there are caveats that come with any new technology. One of the caveats here is that eDNA analysis, because we target minute amount of DNA, it is easy to miss detections. So false positives, false negatives are always a concern, as are false positives. Contamination is always a concern, and we have to be incredibly careful in our sample collection. So again, a figure from that recent review, the use of eDNA really is exploding across the globe. This just shows eDNA studies that have been done in the last decade, essentially right across the world. And we see that there's obviously more in North America, Europe, Asia, but we see a nice representation across everywhere. And the different colors just represent the main two types, I would think, of eDNA methodology. And I'll talk about those next, one being metabarcoding and the other being species-specific qPCR. The other interesting thing that was noted in that review was that we do see this focus on aquatic fish species. And we can see here, if we just look at the to the right of this, is just summarizing the publications that have come out between 2012 and 2021. And we see that a large portion of them are freshwater fish or marine or anadromous fish. So eDNA is rapidly being adopted across the globe, across multiple systems, to explore changes in species, community distribution, abundance, and aquatic fish species are well represented in published studies and well suited to eDNA application. So Atlantic salmon falls soundly in that category. eDNA is also a very effective tool for monitoring changes in species distributions and new invasions. And two that we work on in my lab is pink salmon and increasingly striped bass. So pink salmon, we know, are kind of exploding across the North Atlantic, showing up in rivers all the way from Russia through the UK, Norway, Iceland, Greenland, and down to Labrador. And of course, our ability to monitor for the extent of this invasion is limited. We don't have monitoring facilities on a lot of rivers, so eDNA is a really useful tool to detect pink salmon when they show up in rivers. And similarly with striped bass, we know striped bass are moving northward in Atlantic Canada. We're seeing them increasingly in southern Labrador, and the ability to detect them in rivers using eDNA really provides, again, a super powerful approach. The other really attractive thing about eDNA is because we are essentially sampling water, either filtering in the field or filtering back in the lab, it is relatively low tech in terms of sample collection, which makes it very amenable to community-based sampling programs. And we see that all the way from across the North Atlantic to Africa to you name it. There are places doing sort of community-led eDNA sampling projects, and we have one starting this summer across the coast of Labrador. So very powerful approach, putting science in the hands of citizens, collecting samples and working in collaboration with scientists. So the standard workflow looks something like this. And as I said, step one is water, either water collection and filtration or collection and bringing back to the lab. Generally, we like to filter in the field. So it would be collection and filtering. The filter membrane will be taken, and that will be sort of the DNA attached to that filter membrane will be extracted. And then that would be either sequenced, the DNA would be extracted, that would either be sequenced, or sort of the species-specific DNA present would be quantified. And then, of course, data analysis and interpretation. So pretty simple workflow. We're just sampling water, we're filtering it, and then we're sort of quantifying the amount of species, either species-specific or multi-species DNA present. And sampling looks essentially like this. It can be either done sort of high-tech with eDNA design specific backpack units that filter. It could be done with a water bottle, or there are more sort of low-tech citizen science-based sampling units. And then, as I said, the filters are removed, preserved, and then brought back to the lab for analysis. So there are two types of analysis that we tend to do, either metabarcoding or qPCR. And they differ both in cost and sort of breadth of taxonomic coverage. Metabarcoding use sort of conserved, often mitochondrial markers that sort of allow us to detect multiple species. And this could be a really broad suite of species, everything from bacteria to whales. Oftentimes we focus on marker sets that might provide great coverage or good coverage for a discrete set, such as marine fish, but using things like CO1, we can really get broad coverage. So it's a very powerful approach to get sort of ecosystem level resolution very easily and quickly. The single species approach here, which is quantitative PCR or qPCR, looks at a marker that's been designed to quantify the amount of DNA present from a single species. And that often provides more sort of accurate estimates of the amount of DNA present from that species, and it's often cheaper than metabarcoding. So for certain circumstances where we're looking at, an invasive or targeted work on Atlantic salmon, qPCR can really make sense. And we use a combination of both in the work we do in my lab. So to get into some examples here, I'll just touch on what I think were some of the nice comparisons that have come out over the last, 5 or 10 years, and then I'll get into some of the work that we've been doing. A lot of work has centered on comparing eDNA approaches to traditional survey methods. And this is one of those earlier papers. This paper here compared eDNA-based detections of species to, a suite of traditional measures, be it fish pots, angling, push nets, spike nets, multi-mesh gill nets, snorkeling, beach sailing, bottom trawling, you name it. And what we see is that eDNA provided sort of some of the best estimates of diversity compared to these other methods. Night snorkeling came close, but eDNA really did a good job. And I think it highlights the fact that all sampling methods will have bias associated with them. They'll have catchability issues. There'll be challenges associated with detection of certain life histories. eDNA really does a good job of capturing a broad suite of species independent of their life history or habitat. The other interesting thing that this paper did was it did some experimental work to look at, well, how long can we detect the eDNA from a species after it's been in the environment? And this has been replicated other times, but it just gives us a nice time frame to say, if we get a detection, what does that actually mean? And they simulated this using DNA from 2 species, and they showed that the DNA essentially decayed on the scale of, days to a week. It wasn't persisting in the environment for significantly longer than that. So if we get a detection, that's sort of the temporal scale that we're talking about. Again, other studies have sort of looked at the comparison between traditional methods and eDNA in different environments. So there was a paper on sort of deep sea fishes, again showing this, just as Venn diagram here, showing a lot of significant overlap between the two methods. Another study here looking at overlap of detections in an urban estuary environment. This is the Hudson River estuary in New York. And they saw, again, a really nice temporal overlap between eDNA and seining surveys for different species, anchovy, hog choker, and striped bass. So again, suggesting that eDNA can capture the diversity of species present, but it can also do a really good job of capturing temporal changes seasonally of species as they move through the environment. So in addition to just capturing identifications or detections and diversity, the other question we get asked a lot is, well, what about abundance? How does eDNA do in terms of making some or containing some signal of the number of individuals present in the environment? And this is something that's been looked at for over a decade now, essentially, and it's been tackled in a variety of approaches. The earliest approaches essentially looked at fish in tanks. The Kelly et al. Paper used the Monterey Bay Aquarium and just looked at the proportion of the biomass of fish in the aquarium to the proportion of DNA they could detect from the water, and they saw a positive association. So, and again, so that's kind of the first hints that there might be something here in terms of abundance estimation based on the amount of eDNA we're seeing. Now, there's been subsequent studies like this Evans et al. In 2016 paper that looked at using mesocosm, so essentially big fish tanks, and looked at how well, if they put different numbers of fish in there, how well the number of fish or the biomass corresponded to the number of reads they're getting from the eDNA sequencing. And in all cases, they see, well, in most cases, except for one, they see a positive association. The fits are not great, but there is a positive association. So with increasing biomass, we see increasing amounts of eDNA. And I think that's, as we'll see, that's kind of the take home from the abundance angle, at least so far. The abundance question is that there is a positive association, although the fit is not always great, and it's a little noisy. So it's difficult to do something quantitative, but it's certainly, there is a signal there of abundance. And this is a recent paper which I think continues to drive that home. This is a trawl and eDNA comparison from a coastal survey in New Jersey. And it basically, all that's doing here, this is metabarcoding, and it's comparing metabarcoding of eDNA samples taken before and after the trawl to biomass of fish or surface area, total surface area of the fish caught in the trawl. And we see that there's a nice association. So each point here is a different species at a different survey point. They had 4 seasonal surveys, but really nice, pretty nice relationship for how biomass correlates with eDNA reads. And there is some improvement as we start to look at sort of a more allometric approach to this using percent surface area. But in both cases, we see a really nice relationship. So again, I think the answer here is that there is a pretty strong signal of abundance in eDNA data, with the caveat that it can be noisy and there may be other things that we need to consider. Here is a paper that uses eDNA on Pacific salmon. So this is using eDNA through the spawning season of sockeye and looking at how well changes in eDNA correlate with abundance of sockeye, either live or dead, in the river system. So the lines here, the solid line represents the number of live sockeye moving into the system. Dashed line is dead, and then the eDNA numbers are the gray. And we do see an association. Pacific salmon's a little bit different, of course, from the Atlantic scenario, because we do have death after spawning, and that seems to be a significant driver of eDNA, as you might expect, but there is an association here, and that's encouraging for work on Atlantic salmon. So just in terms of finishing off some of the examples from the literature, I have a few last bits and pieces that are kind of interesting other applications, I'm calling them. And I think it highlights how eDNA can be applied in a lot of different ways to help inform fish conservation and management. This is a study where they use the comparison of mitochondrial DNA and nuclear DNA. So mitochondrial DNA coming from the mitochondria, nuclear DNA in this case would be coming from gametes, most likely sperm probably, but the comparison then of the ratio of the two indicated when spawning was happening. So they sampled, over a sampling period, and they could see this bump in nuclear DNA, and that was associated with spawning period. So we can use eDNA, at least in this context, to identify when fish are spawning in a river system or a water body, which is a nice application. We can also use sort of interstitial DNA from reds to identify the species that is incubating within the red. And this is a study, again, from Pacific salmon, but the principle should hold that interstitial water taken from between the cobble or the rocks and the red should contain elevated eDNA from the species that's incubating there. So they were able to correctly identify the species that was incubating in reds using environmental DNA. The last one here that I'll highlight is another kind of more recent application, but kind of a really cool one. This is where instead of using DNA, they're using RNA, which is expressed DNA. So because they're using environmental RNA, they're able to target genes that are being expressed that may be associated with different processes. So it could be associated with maturation, with reproduction, with different life stages. So the potential now exists to identify using eRNA differences among individuals of different life stages. So in this paper, they were able to identify breeding populations or juvenile individuals and distinguish between the two. So they're able to distinguish between life stages in an amphibian, but again, there's no reason why this couldn't work with fish species such as Atlantic salmon. So the ability to focus on environmental RNA, which is again expressed and should be able to resolve differences between life stages or reproductive status, things like that, really opens up the opportunity for new application. Okay, so that is kind of the end of the literature stuff. We're seeing, and I'll get into our examples next, but I just want to highlight that we are seeing a lot of applications on Atlantic salmon. And this is just a bunch of papers that I pulled from the literature showing eDNA being used to Atlantic salmon. So I know the foundation is increasingly getting applications or proposals to use eDNA for Atlantic salmon, and that makes sense. We're seeing a really a good uptake of the technology by folks working on Atlantic salmon, and for good reason. So I've got 3 or 4 projects. The first 1 or 2 are a little bit longer, and then the last 2 are relatively short, that I'll just highlight how we're using it in my lab. The first is part of a PhD by Sammy Crowley, shown here. What Sammy was doing for her first chapter was using environmental DNA sampled across Newfoundland and Labrador to look at patterns of freshwater fish community composition. And then by comparison with traditional data on fish distributions, try to make some inferences in terms of how things might be changing or how they might have changed, or which methods might be more powerful. And her work was recently published in Environmental DNA, and you can see the reference there. So this is what her sampling looked like. Not sampled, different regions were sampled in different years, and some of that had to do with the pandemic. But Labrador was sampled in 2 years, and we sampled Newfoundland. Extensive sampling, as you can see, lots of samples around. We sampled almost every watershed that we could get access to. Metabarcoding, so this is multi-species community composition work using 3 different markers. So again, highlighting, sort of maximizing our ability to resolve fish composition, but also more the broad diversity present, fish community composition, but also broad diversity present. And I'm not going to say much about the methods other than to say that we spend a lot of time thinking about how to balance controlling for contamination and detection probability, detection power, and identifying high certainty detections. So this is what we see in the detections that Sammy got. And this says preliminary, but that's an old slide. These are essentially published now. So she saw, 30 plus species could be detected. A lot of the ones we would expect. And the panel on the left just shows sort of the mean depth, the number of reads we were getting for each of the different species. It's interesting, we were seeing a couple of hits of marine fish, which was a little surprising. These samples were taken above the head of the tide in river systems, and that could be due to otters bringing an Atlantic cod in to eat, or a seabird or something. So they weren't super common, so that probably explains our detections. But again, the usual suspects, rainbow smelt, American eel, suckers, sticklebacks, Atlantic salmon, Arctic char, the sort of the usual suspects. When we look at community composition, the analysis really separates kind of the fish community, freshwater fish community on the island and sort of southern Labrador from what we see in sort of Lake Melville and northern Labrador. And we could kind of identify two biogeographic groups. And those are shown there in yellow and blue. The plot on the right shows community composition, sort of species richness. So essentially where we're seeing the most species. And perhaps a surprise, Lake Melville really was a hotspot. That's where we see the most diversity in freshwater fish composition or species richness. It really is around the Lake Melville area. The only other pattern really we see is that as we move up into the Torngats in northern Labrador, species richness drops. And essentially what we're getting up there is, Arctic char and sticklebacks. And again, that's not a surprise. But the take home was that communities were strongly influenced by spatially structured environmental variation. When we compared it to the traditional data, so we had, in collaboration with the province, we had access to a large environmental or fish detection data set that was pre-1987. And we did see some interesting patterns. There was significant variation that could be explained by data source as well as climate. So there was some evidence for climate changes in the distribution of some of these species over time, but there was a lot of variation that was confounded between the two data sets, of course. We couldn't separate just because of the way the environment is structured in this system. When we look at sort of losses and gains, so each of these points represents a sampling location and whether or not we saw a loss of species or a gain, generally we see more sites gained species with eDNA versus historical methods. And the historical methods would be gill netting, fyke netting, things like that. So it looks like eDNA is, and as expected, a much more sensitive sampling method. And there is evidence of sort of climate-associated changes over time, but as I said, a lot of the variation here was confounded just due to the nature of the system. Some interesting patterns that did jump out that would be of interest to folks, I think, is that there were some species that we were detecting that we may not have detected. The first one would be pink salmon. So as I mentioned earlier, we have been seeing pink salmon explode across the North Atlantic, and primarily in odd years because it's an odd year population, and we are detecting them throughout Labrador. So the points here, the brown points, show detections through our eDNA data set of pink salmon showing up in Labrador. So that would be either 2019 or 2021. Another interesting one was European eel. So we really didn't expect to see European eel show up. Certainly American eel is widespread, but not, we didn't expect to see European eel. And then what's interesting here, so we look at the detections of European eel and it's primarily in southern Newfoundland. So it's not only is it unexpected, but it's really localized to a very discrete region of the survey. Which was kind of fascinating. And I'll talk a little bit about that next, because we did look into that a little bit further. The other interesting thing here is looking at occurrences of Atlantic salmon. So we do see detections of Atlantic salmon, of course, everywhere throughout Newfoundland and Labrador, but we're seeing them more further north than we might have thought. So that's where that yellow or gold box is. And that could be consistent with range expansion, certainly consistent with the anecdotal reports of salmon showing up, in more northern areas on Baffin Island and in the Torngat. So that's consistent with that sort of pattern. And those would suggest that we really need to start looking at these places to see if we are seeing the beginnings of a range expansion in the species. So for the American or the European eel stuff, we were really fascinated by that. So we did go into some of these systems and sample eel and then sequence their genomes to try and understand if those European detections were real or not. And unfortunately, we couldn't get into all the systems where we had European eel eDNA detected, but we did get into a handful. And then we used some existing sequence data for North American and European eel that we could evaluate or assign individuals back to. And what we found was that we did find evidence of individuals that we had 3 individuals that had European mitochondrial DNA. So fit with what we were seeing in the eDNA, and they showed up in a site in Placentia Bay. And all of these individuals, as I said, had European mitochondrial DNA, and they had intermediate nuclear DNA, which suggested they were some kind of hybrid, hybridized individual. When we look at identifying what they are, they look like they're an F1, North American, European, and 2 back-crossed individuals. So this is not new to science. There's good evidence that European and North American or American eel hybridize and show up in Iceland, but to our knowledge, they had not been detected certainly at sort of a regional level in North America. So this is kind of a new occurrence, and why it's only showing up in southern Newfoundland is fascinating. It could be something to do with dispersal of the larvae, but a really neat sort of a new natural history story that probably wouldn't have been detected had it not been for eDNA. So example 2, this is work that we're doing now evaluating, similar to that Pacific salmon study that I talked about, evaluating how eDNA sampled in rivers can reflect spawning runs of Atlantic salmon. So as many people will know, Atlantic salmon throughout Newfoundland and Labrador are usually counted or assessed using counting fences like this one shown here in this aerial shot, where salmon are directed into the trap, they are counted, or using a video camera are counted, and then they proceed upstream. Of course, these are difficult to maintain, that we have washouts increasingly with high water conditions and accumulation of vegetation on the fences. So we're just looking at other ways to potentially try and add some value and potentially assess in regions where counting fences might not be possible. So we looked at 3 counting fences across Newfoundland and Labrador: Western Arm Brook, Campbellton, and Garnish. And we had sort of staff who were working on the fences take water samples for eDNA every 2 days throughout the spawning season, or sorry, throughout the run in both, in 2 years, 2021 and 2022, I believe. And we also took, all the standard environmental data, and then we conducted. This is an example where we looked at species-specific qPCR. So we were really only interested in Atlantic salmon. So we were able to do a species-specific assay and then look at how it matched with the counts that had been moving through. And the first thing we noticed was that if we just look at the total amount of eDNA detected at each river versus the total salmon count for each river, we do get a positive association. And that's what's shown here. So there is some evidence that total amount of DNA detected during the spawning run does match with the total salmon moving through the system. And for 2 of the 3 rivers, the changes between years did match the direction of the change in the salmon count. So for Campbellton, we see this change here, and sort of an increase, and then for Western Arm Brook, interestingly, Garnish did not, and Garnish sort of bucks that trend, but yeah, so there is this positive association. But then the next question, of course, is, well, what about across the run? So here are the salmon runs. The abundances are shown in the salmon moving through these counting fences are shown in the dashed line. The eDNA is shown in the shaded area, and we can see that there are some matches, but they're not perfect. And I've ordered these by decreasing run size. So the biggest run being on Campbellton is at the top, and that's kind of where we see the best match. And as we go to smaller run size, we see more of a mismatch. And in fact, if we look down here at Garnish River, we see eDNA is elevated right throughout the run, even though salmon, the returning adults, are really focused here. And the same thing in 2022. So the obvious explanation for this is that what we're detecting is, background DNA from either juveniles or Wananish or landlocked salmon in these systems, and that when we have smaller runs, separating the noise, the background noise from the anadromous signal, can be more challenging. So we've looked at correlating the eDNA to the run data, and we looked at various lags. So, a lag of 1 day, 5 days, sort of different ways to do it. And what we see is that a lag of 1 day generally gives us the best correlation, and that makes sense. So the count of fish that moved through the trap yesterday, and those would be just above the counting fence, corresponded best to the eDNA collected just above the counting fence. So that's not really a shocker. Just looking at the strength of the correlations, for Campbellton Campbellton is really where we see the best correlation, and we had that 1 year where we had a fair number of salmon move through, more than closer to 4,000, and that's where we get a correlation that's close to 60%. When we have smaller counts, we see less strong correlations. And I think that's really just comes back to what I said. It comes back to separating the noise from the signal. I guess the take home from this was that there is this association between eDNA and count in terms of reflecting the run. We really need to work on separating the background noise from the anadromous signal, and there are things we can do to try and to do that. One will be looking at RNA to focus on, something that might be life stage specific, and the other is right now we're working at using larger targets. So as DNA ages, eDNA ages, it gets broken up. So if we were to look at larger fragments of DNA in the environment, maybe that would give us a more immediate reflection of fish moving through it. And we're kind of looking at both of those things now. So that's a little bit of a work in progress. And then the last two things I'll talk about are a little bit shorter, but I think they'll be of interest. The first is this project where we're pairing eDNA with telemetry work in southern Newfoundland for Atlantic salmon. And the goal here is to sort of really leverage the ability of eDNA to capture, a broad suite of species and really the diversity in the ecosystem to help us understand what environment outmigrating smolts might be experiencing. So here we're combining a acoustic telemetry project where folks are tagging outmigrating smolts from the Conn River. They tagged them last spring, and then they were tracked using this network of receivers right throughout through the Bay d'Espoir. So we know where the smolts were and how long they spent in different places and where they moved around. But then we're overlaying on that this eDNA survey. So this is the eDNA samples collected while the smolts were in the environment last summer. And we're currently analyzing these samples, but what this will give us is the ability to identify, were there significant predators in the area? What did the prey field look like? All these sorts of questions that we really can't, it's difficult to get at other ways. And here with water samples taken relatively easily, we can start to piece together the picture. So I think the combination of the two here could be really powerful to help understand what's going on with Atlantic salmon in the environment. And this is just what our sampling looked like. We were sampling very low tech, just using a Niskin bottle, taking 8 liters of water at 4 meters depth. We had hoped to do deeper, but there was a hauler accident and we were unable to use haulers at the time. So 4 meters depth was really where we expect salmon to be anyway, our smolts to be. And now we're in the process of metabarcoding these samples, again with 3 markers to really get at the diversity of fish species or other taxa present. Okay, so then the last thing I'll talk about is, this is part of the master's project of Sam Beal, and this is not on Atlantic salmon, it's actually on Atlantic whitefish, but I think it's relevant for a couple of things that will hopefully be clear. For those who don't know, Atlantic whitefish is this endangered whitefish. It's only found in a couple of locations in Nova Scotia. It's really on the edge of extinction. So it makes a really nice test case for improving or testing the sensitivity of eDNA to detect very rare species. And to that end, whitefish have been very difficult to detect with traditional measures. And there are a lot of folks at DFO who spend a lot more time than I do thinking about how to detect them and their recovery. So what Sam did for her master's was to ask the question, well, are there other markers, other portions of the genome that might provide more sensitive measures of whitefish DNA in the environment? So we weren't really having great luck with mitochondrial DNA. So she set out to identify other markers that might increase our sensitivity. So she settled on something that's called a single short interdispersed nuclear element or sign. And these are found throughout the genome. They're non-coding, they're relatively short, 100 to 500 base pairs. They're mobile and highly repetitive. And these have a history of being used in forensic applications. What makes them really useful here is that they're high copy number. So there's a lot of copies of this across the genome, which sort of should increase our ability to detect them in the environment. However, to our knowledge, again, prior to this study, they hadn't been used in eDNA applications. So there might be some significant advantages over mitochondrial DNA. So Sam did a bunch of stuff, but I'll just highlight one portion of her thesis here. And this is where she held whitefish in a cage in a lake in Nova Scotia. and then looked at distance from the cage and looked at how the detection of the mitochondrial or the sign changed. And that's what's shown here. So this is distance from pen, and this is the amount of eDNA present, and the sign or this nuclear marker is in sort of this green, blue, and red is a mitochondrial marker. And the first thing we see is that we get significant improvement of detection with this new marker. So we do a lot better in terms of detecting the sign than we do with mitochondrial markers. And then as we move away from the pen, by 20 meters away from the pen, we start to lose detection with the mitochondrial marker. And that's a question that folks might have as well, is that how far do we have to be from an organism to detect it? And in this case, in this lake, it was 20 meters. However, with the sign, we didn't lose detection even up to 80 meters. So it really suggests that this field really is moving quickly, and we can go a long way perhaps with exploring other types of markers to improve our sensitivity and our detection probability. And that's going to be really important in species that are endangered and at really low abundance in the environment. So that's really all I have. Now I have a few slides to summarize, and then I think we should have some good time for questions. Targeted approaches such as qPCR detect and potentially quantify the DNA of a single species and use single species primers. So that's something that would be super relevant for Atlantic salmon or sort of aquatic invasive species. And that really contrasts this sort of metabarcoding approach, multi-species, which tends to cost a little bit more, but gives us more sort of taxonomic diversity and allows us to, in many cases, to start to reconstruct the environment or the ecosystem that's there. And for from the Atlantic salmon perspective, the ability to identify prey and predator species present could be, as I said, very, very powerful. And that, I think, for both of these reasons, eDNA analysis is likely to be central to the management and conservation of Atlantic salmon in the coming years. It's really good at detecting the presence of species in the environment, and it does seem to do a better job over traditional methods like gill netting and fyke netting and things. And I think that's only likely to improve with things like the application of different marker type signs and different ways and increased sequencing capacity. That's going to be really powerful in the realm of climate change when we have shifting distribution patterns. Pink salmon and striped bass in Atlantic Canada are only going to be the tip of the iceberg. We're going to see a lot of changing distribution patterns, and our ability to understand what's where and where things are moving is going to be critical to the conservation and management of fish species. So I think what we're really seeing is the beginning here of the use of this technology in that application. And again, as I've said already, I think the ability to resolve community composition and changes over time that might indicate, key prey and predator species, but then integrating those with telemetry data is going to be really powerful. That's going to tell us a lot about what salmon are experiencing in the natural environment. In terms of abundance, of course, this is always a question that comes up. I think it's becoming clear that there are associations between abundance and eDNA present. We see it in Atlantic salmon, we see it in Pacific salmon, we see it in marine fish. The challenge, of course, is always that these relationships are not always super strong. They can be noisy, and we need further consideration of how we might sort of distinguish noise from signal in this context. Some of that could be using eRNA or targeted marker design to sort of separate life stages or older versus recent DNA, or consideration of conditions. Flow and temperature can impact the ability of eDNA to degrade or to persist in the system. Nonetheless, I think it's clear that eDNA will provide a powerful indicator of changes in relative abundance. And there's been some work within DFO, I think, most recently that kind of demonstrates that. So that's what I have. I am happy to take any questions. And if anything was unclear, please don't hesitate to send me an email. I'm happy to follow up.
Well, thanks, Ian. That was a great presentation, and a lot of information there on eDNA and this new tool that we start seeing more and more. We do have time for questions and answers. So for those that joined us before, you can raise your hand or type in. The preference would be to raise your hand so you can ask your own questions. So that way I don't make any errors when I'm trying to read and it comes out a different way.
Are you there?
Yeah, I'm here. Can you hear me, Ian?
Oh, there you are. Sorry.
Okay, so you didn't hear me. I'm sorry for that. I was unmuted, but So we do have a good 15 minutes for questions. It could be by raising your hand, so you can ask your question yourself, and that would be the preference. But if your computer, your system doesn't allow you to do that, you can type it in, and I'll do my best to read it out loud for Ian, and he'll be able to answer it. So we are open now. I'm just going to check. We do have some hands raised. So we're going to start with Nicholas Lapointe. So you are unmuted.
First, great presentation, Ian. Thank you. Somewhat unrelated question. I'm just curious about the potential for eDNA and whether there would be any possibility in using it to assess, say, historic distributions of something like Atlantic salmon above a barrier that's existed for 100 years or more. I know environmental DNA would degrade over time, but would any, be preserved in sediment cores or something where that might be detectable? Has anyone looked at that?
Yeah, I don't know if anyone's looked at that for Atlantic salmon, but certainly there's good precedent for doing that. And I know folks have done that in other species. So using sediment cores to reconstruct the presence and distribution of species is certainly possible. And that would be a great thing to try and understand, previous distribution patterns, especially in the context of climate change impacts. Super powerful.
Okay, thank you. Robert Lennox, you can ask your question. Go ahead.
Okay, now I'm unmuted.
Okay.
Perfect. Great presentation, Ian. Thank you very much. Really interested in the pink salmon. Curious what kind of, I don't know if you had multiple samples across years and if you see any spatial or temporal spread, or if you have any other comments about the pink salmon program that you're working on.
Yeah, so we've been seeing them since, I don't know, 2017, I guess. And up until last year, the numbers had generally been increasing. So going from, 2 or 3 to 5 or 6 to 10. And we had expected last year to continue that trend, but for some reason it didn't. I'm not sure why. We do have a project just funded it's sort of developing a community-led monitoring program throughout Labrador. So we're going to be up there for the next 3 years. So we hope to continue that monitoring stuff. So it'll be really exciting to see if we continue to see detections. The patterns last year elsewhere were certainly consistent with what we'd observed previously. Numbers continue to climb. So last year was a weird year environmentally. I wonder if they were just later. We didn't overlap with the fishery as much, which is generally where we get our detections. Who knows? But yeah, it'll be super interesting to follow.
Do you have any sense if these are self-recruited pink salmon or if they're all straying over from the rivers in Norway?
Yeah, we don't know. One of the things we're going to do with this project coming up is go into a lot of these systems, hopefully in the early spring, and collect eDNA. So that'll hopefully answer that question. My gut feeling is that we're still early in the invasion, so we're probably not seeing a lot of recruitment. But who knows? Well, it'll be curious to see if we see any pink salmon DNA in the spring, which would be indicative of juveniles or eggs. So.
Yeah, it's going to be crazy. I've seen firsthand it'll go from a trickle like this, and maybe not next year or maybe not 2 years, but within a few years, they're going to be everywhere.
Wow.
Yeah, they are an extremely adaptable, explosive species. It'll be. I just wonder how long it's going to take. Thanks, Ian.
Yep. No, thanks.
Okay, we're going to move on to Elizabeth Barlow. You can ask your question.
Hi, thanks, Ian. That was excellent. Great presentation. As you know, we're doing some eel work in Manka and Conn River area, and we are looking at trying to develop like a way to be able to determine abundance by eDNA, as you discussed. But I'm wondering if you have the option of using metabarcoding or qPCR when you're trying to determine abundance. Is there a preference for using one over the other?
Yeah, it's an interesting question. I think the consensus in the field seems to be the qPCR may be more an accurate measure of single species eDNA present in the environment. And when we've compared both measures in Labrador, we see an association, but it's not a perfect association between read depth from metabarcoding and qPCR. So that's not a perfect relationship. So there is some noise there. Of course, we don't have the gold standard of knowing exactly how many fish were present in those systems to know which one is closer. But I guess if I was betting, as I said, I think the consensus seems to be that qPCR is probably going to be a little bit of a more accurate measure of eDNA present. Yeah, but I don't think we have a definitive answer yet.
Okay, thank you.
Okay, thank you. I'm going to read the next question. The question is, can one sample be used for species presence/absence, like life stages and abundance analysis, or would multiple samples be preferred?
Yeah, so I mean, you can do, oftentimes we get more than enough DNA from a single sample to do multiple things. So we would do metabarcoding, we do qPCR, we often do different markers for the metabarcoding. So there's lots of DNA usually present to do multiple things. What I mean, often, what I didn't, maybe I didn't say, but we often, of course, replication is key. So we do want to make sure we get lots of replicates. These days we're using 3 or 4 replicates and a blank from our sample. So just to make sure that we're capturing the error associated with our sampling, but you can, yeah, you can use samples, an individual sample can be used for multiple purposes for sure.
Another written question, and it is just wondering, DNA analyzed using metagenomic community approach was not considered. So like a question/comment. Maybe it was part of your presentation a while ago.
Yeah, I'm not sure if I catch that, but if the person wants to follow up, I'm happy to chat about it.
Sounds. Okay, just looking for other questions. Well, it looks like these are the, oh no, we got a hand raised, just one question, just 1 minute. Mike Rush, you can go ahead.
Oh, thank you. Hey Ian, this is Mike from Kushabuguak Watershed Association. I was just wondering, like, what's your feeling on how reliable eDNA could be for life stage analysis? Like specific examples, like, you know, we're curious, there's striped bass spawning in our rivers. So, you know, you could look at different life stages, or even kind of one thing that hasn't been mentioned in this presentation, I guess, is like brook floaters is another freshwater mussel we deal with, and we're one of the few rivers that still has them. Yep. It'd be nice to know, like, get a sense of like recruitment levels and stuff like that. I just was wondering your opinion on that.
Yeah, I mean, in terms of spawning, as I said, the comparison of the nuclear DNA to the mitochondrial DNA can be a useful tool to denote spawning if it's happening. The eRNA stuff is really at the beginning, I think. So it's it looks really interesting and really cool. I'm just not sure at the moment how far we're going to be able to take it, but it's certainly something that a lot of us are thinking about testing and sort of kicking it around to see how well we can use it to answer those exact questions. So I think for us, trying to, in Newfoundland in particular, trying to separate, you know, returning adults from juveniles in that context that's something we want to try. And if there are other test cases, yeah, I mean, I really think we need to really kick it around to see how far we can push it. But it's certainly very encouraging based on those first couple of papers.
Thank you. Good presentation, by the way.
Thanks.
Okay. I think that might be our last question, but like Ian said, if there's follow-up questions that you think of afterwards, please reach out. I believe his email address was at the start of the presentation. If you didn't write it down, please contact me and I'll be happy to share it with you. Just want to say, just thank you, Ian, for being available today. I know you're really busy, and to be able to share your knowledge on eDNA, and it's a really great way to finish our 2023-2024 series. As you know, usually our webinar series start in September and we go on to March, April. So we're already starting to collect topics and potential speakers for next series. So please feel free to reach out to the staff here at the foundation if you have any ideas of something you would like to learn more on or hear someone speak. We would appreciate that. So once again, thank you. Thanks for all the speakers this series, and also thanks for all the participants that registered and joined us today. And I just wish you all well. Thank you. Thanks again. Oh, and it's recorded, so you can see it on YouTube after too, for those that missed it. Forgot to mention that. All right, see everybody.